name is Faraz Ahmed, and I'm currently a senior at NYU Polytechnic School of Engineering. And today I'm here to talk about how the change in pH in a solution can result into a determining factor for DNA sequencing. So the content of my presentation is that I'll just talk a little bit about the DNA anatomy. I'll talk about the nucleotide addition, and I'll also talk about the methods that I'm implementing in my research in order to detect the nucleotide addition. And I'll go over briefly how I'm preparing my materials and then results and future work. So DNA anatomy, just to go briefly over it, we all know DNA is a double helix molecule. The backbone is basically a repeated unit of nucleotides. And it's anti-parallel, which just means like if we compare one strand with another strand, one of the strands is going to seem to be 180-degree flipped over the horizontal axis. And both of these strands are held together by hydrogen bonds. And the anatomy of a nucleotide consists of nitrogenous base with a 5-carbon sugar in the oxyribose and a phosphate group. The nitrogenous bases are actually adenine, guanine, or cytosine and thymine. This is just to show that cytosine and guanine can actually form 3 hydrogen bonds, whereas thymine and adenine can only form 2. So actually in vivo, there are multiple enzymes that are used in order to carry out the synthesis of DNA. But one of the major enzymes is the DNA polymerase enzyme. And as you can see here that the nucleotide addition is actually occurring at the 3' end. So a new nucleotide is incorporated at the 3' OH of the ribosome, the ribose sugar, I'm sorry. And over here you can see that this is the OH group, and when a new nucleotide is incorporated, from this OH group a proton is lost. So this is a very key point because our detection is basically focusing on that proton loss, and I'll get back to that. And in vitro, obviously, we have polymerase chain reaction. We have 3 steps in it: denaturation, which is basically melting of DNA, and it separates the 2 strands. Then we have different primers that anneal the specific regions of the DNA sequence when the temperature is lower to about 45 degrees Celsius. And after that, if we raise the temperature at 72 degrees Celsius, the DNA polymerase comes in and actually incorporates the DNTPs inside the reaction. So now I'm going to talk about the implemented methods. So how do we detect the proton loss? We are currently using conductive polymers, and particularly in my case I'm using polyaniline, and it belongs to a semi-flexible rod polymer family. And under appropriate oxidative and doping conditions, it becomes highly conductive. And since we are dealing with PCR conditions, so we are manipulating the temperature, so it's very good to use the property of honey as a thermal stability polymer as well. It exists in 3 states, but the state that we are focusing on is the emeraldine salt form, mainly because that form has conjugated bonds, which allows it to be conductive. And in order to do that, we are using open circuit potentiometry, and we are using drop-sense technology. We're using drop-sense electrodes. And we are currently focusing on just the working electrode, which is the dot in the middle, and the reference electrode. The working electrode is where we drop cast the polyaniline. We have polyaniline that is doped with dinyl naphthalene sulfonic acid, which is DNNS8. And we have it as a stock in 44 weight % concentration, so we sort of dilute it down to 1 weight % in chloroform. And once we do that, we put 8 microliters on the working electrode and let it evaporate at room temperature. Since it's chloroform, it evaporates really fast, and once it evaporates, it creates a homogeneous layer of polyaniline at our working electrode surface. And since when you actually dope polyaniline, you have to use excess acid. So when polyaniline is doped, it also has like multiple protons that are just there, which are not part of the polymer itself. So in order to remove those protons, we use 100% ethanol, and you have to just rinse it for at least 2 minutes just to create a homogeneous layer of polyaniline. And once we do that, we have to incorporate one of the organic solvents, which is glutaraldehyde. Glutaraldehyde covalently binds to polyaniline, and once we have that sort of sandwich, then we can actually introduce our Taq polymerase enzyme, which comes from Thermus aquaticus and used in the PCR technology, and that can also bind to the glutaraldehyde. So on our electrode, we have the polyaniline layer, then we have the glutaraldehyde, which is binding to the polyaniline, and now the same glutaraldehyde, the other end is going to bind to the Taq polymerase enzyme. So now we have enzyme immobilized on our electrode. This is actually the reaction chamber in which the actual reaction occurs, and the potentiometric measurements are done basically by using the EMF reader. So let's talk about results. As you can see, the big peaks are basically just the manual or physical noise that you see, because our system is very sensitive. So as soon as you pipette something on it, it's very sensitive to that pipetting movement or like the gravity, and it creates those peaks. So let's talk about what's going on in this graph. So in the first peak that you see is actually the PCR buffer addition. The PCR buffer is actually has the right amount of salt concentrations and pH, which optimizes the PCR reaction. Next up, the two peaks that you're seeing here, this one and that one, is actually showing the DNA template and the primer addition. And now we have everything. We have the enzyme, we have the DNA, we have the primer. Now we have to incorporate the DNTPs inside our reaction chamber. And keep in mind that it is being done at 72 degrees Celsius, which is the extension of the DNA PCR protocol. So over here we see that we added DCTP, and the level of voltage remained the same as it was before. Same thing happened when we added the thymine into our reaction chamber, and the voltage remained the same. But as soon as we added adenine, we see a little jump from here to here. That is actually due to the loss of protons into our solution, which changes the pH of the solution. Hence the reason why you see the voltage. And this can be further explained by our experiment that was performed in order to study the relationship between pH and the electromotive force, which is recorded by the potentiometer. So you can see that the relation here is inverse, which means that if you decrease the pH, which is happening in our case, the voltage goes higher. So since we have loss of protons, the pH is changing, hence the voltage goes high. So in our future work, as I mentioned earlier, we have very much, we like to do more additions of DNTPs. It's just that the temperature conditions and the environment design is very difficult to achieve. And currently we are using 21.3 nanograms per microliter concentration of DNA, and we would like to go down further to optimize our results. And that's it. These are my references, and thank you for listening. If you have any questions, please.